Simply opening the cell might create enough of a disturbance.
Okay, yeah, that is so interesting.
These Norwegian bees, they have kind of lower levels of deep-worm wing virus, but we are seeing kind of a shift in traits that's kind of exciting. Honeybees are absolutely incredible. The most popular question I get is, what do I do as a beekeeper? How do I start if I want to go treatment-free?
All right, so we are live. First, I just want to say thank you so much for meeting with me today. I really appreciate you taking the time out of your day to talk about Varroa mites.
You're very welcome.
Thank you. But today I have Dr. Melissa Oddie on the podcast, and she's doing a lot of really interesting research with Varroa mites. So let's just get straight into it. Tell us who you are, what you do, and all that.
Right, yeah. So you gave my name. My name is Melissa, and I've been researching Varroa-resistant bees and Varroa resistance for about 8 years now. I started during my PhD. And I think most often I was working on a specific population of buckfast bee in Norway. This was a population developed by a beekeeper in Norway. And he basically, in 1998, I think, decided that he was going to stop treatments. It wasn't a popular decision, and he did lose a lot of hives at the beginning. But with careful learning and adaptation, he built from the stock that he had left and incorporated new stock. And over the span about 10 to 12 years, he got his full stock back that was treatment-free, essentially. No treatments, like not even drone cutting. And the population has been fairly successful to this day. So I was called in about 2014, 2015. That's when I started my PhD, because he had been going on about these bees and no one had really been hearing him, listening to him, or believing him. And so they sent me to kind of figure out what was going on. And that's kind of essentially what I did. So we can talk a little bit more about what those bees are doing. I'm also engaged in a resistance breeding program with another population of bees here in Norway. They're Carnica stock primarily, so carniolan bees, just because we wanted to show that it could be done with another stock. This was not just kind of a fluke or a one-off or special thing about these Norwegian buckfast bees, but it could technically be done with any stock.
Okay, yeah, that is so interesting. So, yeah, go ahead and explain what's going on with the buckfast bees that you've been looking at.
Right. Okay. So the story that I was given when I started was that there was a population of buckfast bees that this beekeeper wasn't treating. He had very low loss rates. They were producing a lot of honey, and nobody really knew what was happening. So what I decided to do is first of all, reading everything I could read about resistance breeding, and I started with the Baton Rouge lab bees, and this is when we get into the Varroa sensitive hygiene, the VSH and the SMR. Those terms, I believe, were coined by that lab, looking for heritable hygienic traits that could be bred in bees. So that was the background that I had, is that VSH was a thing. So that is bees actively detecting mite-infested cells and removing them before the mites have a chance to complete their reproduction. And this reduces the reproductive success in the whole hive, and it controls the population. SMR is the ability of the bees to reduce that reproductive success in the mites in a general sense in the population. That's suppressed mite reproduction. And that can be achieved through Varroa-sensitive hygiene, when you basically remove the brood and stop the reproduction in each individual cell. So that's essentially what I was looking for in the buckfast bees at first. And how I went about that was we designed an experiment where we basically found some control colonies. And then, so we had these about 10 Norwegian hives, this buckfast surviving hives, and about 10 of these Norwegian control hives. And then we went to a different population that we knew had kind of an issue with mites, and we took frames of captive brood from those hives. So there's a population that had nothing to do with either of our test populations, because we want it to be standardized across all of the hives. We took these brood frames that had just been capped, And each donor colony gave 2 frames. One went into a survivor, one went into a control colony. And then we basically kept them there for the capped period until a few days before emergence, and we took them out. And what this did basically was it standardized the mite infestation levels in the brood. It standardized the genetic background of the brood. And only thing that was different were the genetic origins of the worker bees that were going to be affecting this brood or walking on this brood. So before we put them in, we mapped them because we needed to know where the cells were and whether cells had been removed. So once we took them out, we mapped them and photographed them again so that we could track if there's anything that was missing. So that was the general setup. And what it did was it provided a high infestation rate for the buckfast survivors so they could act upon these mites, because normally the mites weren't very frequent in the brood. And this might have been a problem when we wanted to collect data on the mites. They were not there. So the results of that paper were published in the 2017 Peer J paper. The official title escapes me, but I can send you a link. It's open access, so anyone can access and read it. And what we saw was when we looked for VSH, when we looked for brood removal, the brood removal rates were comparable between the buckfast survivors and the control bees that we were using. So they weren't taking brood out at any higher rates, which I thought was a bit odd, because brood removal was kind of and still is kind of the kind of principal theory we have for hygienic behaviors. Like, that's the thing. If bees are going to do something, they're going to do that. So I didn't find it. But the interesting thing is I found a lot of cell recapping, which had been cataloged by the Baton Rouge lab, by Harris and Harbo. And they wrote it down as kind of like a byproduct of VSH, kind of an incomplete form. So the cap had been opened, and then they were about to remove the brood, but something didn't quite get done, and they just capped it back over. But I found this in much higher levels in the Norwegian population, which was really interesting because I couldn't find the brood removal at the same time. It wasn't there. So I've been thinking a lot about recapping. It's possible that it could be influencing reproductive success, although I haven't actually found any solid evidence that uncapping a cell really does reduce reproductive success of mites yet. That's still a work in progress. But because mites are so sensitive to changes in temperature and humidity, and they need very specific conditions for reproduction. Like, as I understand, it's still very difficult to breed mites artificially in a lab because we just can't get the conditions right. We can't mimic the conditions needed in the hive. So it's possible that simply opening the cell might create enough of a disturbance that it might cause harm to the eggs being laid by the Varroa mite. It might inhibit mating. It might just generally disrupt things and limit their reproduction. But I don't have any solid evidence for that. That's just a theory now. But I mean, in truth, a lot of other populations we know of across Europe, in South America, they do actually remove brood, like, completely. And it's probably kind of a more intense Varroa management strategy that is employed where mites are more frequent. You have higher densities of bees. You have warmer climates. In Norway, the climate gets very cold in winter. We have a very long broodless period, and that is not good for mites. So yeah, that's the gist of what was going on with the buckfast bees. So they do have SMR. They do have a much lower reproductive success rate in the mites that they have here. And the mite drops under the boards are consistently low from spring to autumn. On average, the line pretty much doesn't move if you're looking at the number of mites that fall down on your boards. It's about 5 a day.
Oh, wow. Yeah, that's not bad at all. So why is the Western honeybee even so sensitive to mites to begin with? Because there's other, like the Asian honeybees or like Africanized bees, they say to have a little bit more resistance to Varroa mites. So why Western honeybees?
Right. So that's a long story, and it starts, I think, in the beginning of the 20th century, when we were developing honey production in Asia. Someone thought it was a great idea to move Western honeybees over to Asia because the Asian honeybee is very adapted to Varroa, but the colonies generally are small. And they're generally very timid and flighty. So if the colony is disturbed enough, they take off, they abscond. So the whole colony, queen and all, they just leave and go find another home. This can also help managing Varroa as well. So they weren't great for honey production, really skittish bees. So the Western honeybee was brought in, and they were really successful. They did their jobs, but they were put into contact with Apis cerana, the Asian honeybee, and Varroa jacobsoni, which is what we called Varroa before it became a problem. So if you have a novel species that is put into contact with a very similar species, there is a chance that the parasites from those species and the viruses can jump. And that's what the Varroa mite did. It was getting into these European colonies, and at first it couldn't reproduce, but there was some mites with some adaptations that enabled the mite to reproduce on Apis mellifera. And Apis mellifera didn't really have any defenses. So what Apis cerana does usually is if a mite gets into the worker brood, it's detected and removed immediately. So the mites adapted to only use drone brood. But the genes kind of dictating that seemed to be variable, so that when they went on to Apis mellifera, it kind of got rolled back. So they started laying on worker brood, and the Apis mellifera bees, the Western honeybees, didn't have a defense to kind of remove the brood at first. So they just kind of let the mites do whatever they want. And then they spread. And then, of course, some colonies were probably shipped back or shipped to different areas, and the mites spread. And because we had very little control on who moved hives where at that particular period in history, it spread very quickly and very well. Now, that's the first half of the story. The second half of the story are the viruses, which we know. We actually do have some populations of honeybee that have high infestations of Varroa, but they're not dying. And that's because there is a specific form of a virus called deformed wing virus that adapted to use the mite as a vector. So before the deformed wing virus was orally transmitted or contact transmitted, it didn't have a very solid pathway into the bee. So the viral titers in the bees were very low. So the infection levels were very low. It's kind of like a cold. But one strain of this virus basically used the mite as a vector to get into the honeybee's blood or hemolymph directly. And that allowed it to be transferred at much higher rates. So the virus became much more virulent. So if you had high mite loads in autumn when bees need to overwinter, or at critical periods during a dearth when bees needed to stay alive for a long time, they couldn't because their lifespan was shortened because they had to deal with much higher levels of this virus. So we can see the transition of a very diverse branch of deformed wing virus. And then as soon as the mite is introduced, that diversity just kind of narrows into one or two species because these viruses have outcompeted all the other variants. And it's just a virulent strain that gets into your bees.
Oh, wow. That's interesting. I didn't even know that.
Yeah. There's a paper done by Stephen Martin, and he has kind of really good evidence on Hawaii when the mite was just introduced to Hawaii, and he tracked the viruses. The graphs are very interesting, so I can send you a link to that paper too.
Yeah, definitely. Do you know anything about how the bees are fighting deformed wing virus at all, since we're on that subject?
There is a lot of studies. There's a lot of studies investigating it. Viral resistance is one of the theories. I can't speak so much from experience with viruses directly, but what I do know is that these Norwegian bees, they have kind of lower levels of deformed wing virus, and I think it's directly related to the fact that they just have lower levels of the parasite of the Varroa. And it seems like this is a very common resistance trait, is they don't deal with the virus so much as they deal with the parasite and reduce loads so that they have fewer viruses. Now, this might not be true for all populations. I know there are actually a lot of populations out there that are becoming resistant or are resistant to Varroa. A lot of them have not been measured by any scientist that I'm aware of, but I know they're present. And it seems that the traits are very different across the board. They're kind of tailor-made to the specific situation that these bees are adapted to.
Okay. So, just curious, how are the winters then over there in Norway in terms of are they having a lot of die off like we kind of are here in the United States? Since you mentioned there are longer winters there, but they seem to be doing better with mites. So, is that helping them have like a higher percentage rate?
It's so it's interesting. So the percentage of resistant colonies is actually quite small here in Norway for now. It's a growing kind of thing. We have only like a handful of beekeepers that have decided to go treatment free, and a lot of them keep the same stock, this buckfast stock or this carnica stock that we're developing. Um, so the winters, we actually have an advantage here in Norway because the colony densities, so how many colonies there are in a kind of square kilometer radius, is very, very low comparative to the United States. So we don't have that many colonies, which means we don't have a lot of mite spread between colonies. And we have these very long winters. So there are just months and months where there's no brood being produced. So I think they stop brood production in around November, and then they kick off again in something like January, depending on how cold it is.
Okay. Oh yeah, I was experimenting a little bit with brood breaks this last year, and that will make a huge difference with cutting back those mites so they can't reproduce.
Yeah, I saw bits of the video on brood breaks. It's good. We actually have a second brood break in midsummer where we have this dearth, so this just lack of
Mm.
flowers, and the queen actually stops for about 2 weeks, and kind of goes to ground. So we have a lot of new beekeepers calling up saying, "Ah, my colony's queenless. Like I don't know where she went," and we're just like, "Give it a moment. She'll come back."
Yep.
Usually, that's what happens.
Yep. Oh, that's really interesting. Wow, so that actually has probably helped out the treatment-free beekeepers quite a bit.
Yeah, and it's actually made research on resistance breeding fairly easy because stopping treatments, I'm going to say, can be very risky depending on the density of colonies, depending on how many mites, depending on how well you control the mites. But the carniolan bees that we have right now, they're in a valley, and all of the beekeepers have decided to join this program. So they're all reducing treatments or stopping treatments, and they have these adapted bees. So I would say that the operation that I'm running is fairly low risk. And all the beekeepers that are kind of in direct vicinity are aware of it and okay with it. And I'm very sure that this is not going to be the case in all situations. So we have an advantage, and I hope we can put out some really interesting research in the future to kind of advise beekeepers if they want to start with this, how best to do it with a low risk.
Yeah, definitely. So let's see. Have everything, all the questions I had about your papers here on a piece of
I've
paper.
got my notes.
Do you? Because yeah, all of your papers are just so fascinating. So I didn't get
Thank
to go
you.
through all of them. So if there's anything else you want to speak about with like the research you've been doing, yeah, feel free. Just go for it.
Yeah,
Geek out.
the most popular question I get is, what do I do as a beekeeper? How do I start if I want to go treatment free? And I have to say, the first thing that I recommend is don't just stop treatments cold turkey, because depending on where you are, that can have a lot of damaging effects both to your bees and to other people's bees. What I recommend is that you definitely monitor mites. So put the boards down, count mites, and see how your fall is. One count generally doesn't do it for an accurate assessment of how many mites you have in your colony. What you need to do is count. Let's leave the board down for a 7-day period, then count, then put the board back under. And if you're feeling really ambitious, do that a third time. And then take the average of those 3 counts. And that should give you an average daily drop that's accurate, because the more brood there is in a colony, and how when that brood emerges tends to dictate how many mites fall on your boards. If you want to do board counts, if you want to do sugar shake or something like that, that tends to be a little more accurate. Um, but basically, you monitor your hives and see how the mite growth rates are. If you find a colony that tends to have lower rates in general, take a look at that at the brood in there. See if you can find recapping. The nice thing about recapping, and why I keep going on about it, is because it's easy to see, and it tends to be highly correlated with colonies that have the ability to survive Varroa infestations really well. So what you're looking for in recapping is the cell cap at the top may look different. It also may not. But what you can do is you take your hive tool or a small knife and you gently take the cap and turn it over. And if you do that to capped brood that's already started pupation, is when the brood pupates, it spins a little silk cocoon, and it's shiny and it coats the entire interior of the cell. And if a bee has chewed open the cell cap, that shiny coating is not there anymore. So you just see like a dark kind of matte hole of wax, and it's usually quite rough because they don't have a chance to smooth it on the underside of the cap because they can't reach it. So you see this dark hole of wax where they've just kind of patted over the hole that they've made. And yeah, so if you get more used to seeing this, you can actually just take a look at a frame and be like, oh yeah, that's recapped, that's not. If you get used to kind of looking at it, so you can assess it at a glance with a little bit of practice in your hives. And I'd say that's a pretty good way of at least getting an idea that your hive might have what it takes to survive Varroa. But yeah, keep a handle on those mite numbers. Choose a mite threshold that you think is acceptable for your area, because it is very different. And then when you see the colonies getting out of hand, then you treat it, and then you just cross that queen off your breeding list, and don't breed from her, and then requeen her whenever you can. So with a stock that you're sure might be like better at surviving. Start small. I would say if don't start with your whole operation. Just limit risk and start with a few small hives. It's also easier to count and maintain hives like that if you have a small number. I would also say work with other people in your community if they're open to resistance breeding or slow treatment-free beekeeping. Then talk to people, because if you only have 20 hives, but you've got 5 other neighbors that have 10 to 20 hives, then you've got almost 100 hives, and you've got an entire population to select from. And this is basically what we've done with the Carnica population. Is we have a bunch of beekeepers who want to go treatment-free, who have a breeder that's already been treatment-free for a number of years, and we have a good profile of his bees. So we have a subsidy program where they're buying queens and stuff, but all of the colonies have the potential to be chosen as a breeder. So I assess all the colonies based on the data the beekeepers provide me, and I make a list based on a mathematical algorithm that I've created to select the best queens based on Varroa, honey production, and docility. And then I give that list to the queen breeder, and we choose the colonies together. And I mean, so far so good. The data looks fairly good. It hasn't been published yet, unfortunately, so I can't share too much. But we are seeing kind of a shift in traits that's kind of exciting.
Yeah, that is really exciting. It's cool to see a different form of resistance against Varroa in terms of bees that can just uncap the cell, and that will actually disturb Varroa enough versus having to pull that brood out and then get rid of that brood, because that was one of the main concerns for a lot of like commercial beekeepers, is if you have these resistant strains that are technically VSH, then is that going to lower their overall population just because they're killing brood in the process?
Yeah, that's debated. Some people say it's not a problem at all for the hives. Some people, like me, saying there has to be some sort of energetic cost. To add another layer of complexity, I think they change strategies based on the resources they have in the environments, the time of the year. They might go through high energetic cost strategies in spring when they know there's going to be the food resources to replace that. And in autumn, if they have a long winter to go through, they might be a little more conservative with their strategies. And that might be something where recapping may play a role without the removal. So the fact is that we just don't know. But I mean, the cool thing is that bees are going to do whatever they need to survive. And they are very capable of changing it up when they need to, changing strategies. It's not just kind of one blanket thing that they turn on and off. But the interesting is with the recapping, they can turn it off if they don't need it, which has been really, really difficult when you need to measure and assess the recapping level of a hive or the hygienic level of a hive. And when there's no mites, you need to actually provide an active challenge of mites to actually record the level at which they can do this recapping or have these hygienic behaviors. Because if there aren't any mites in the brood, there's no reason for them to actually show you that they can do it.
Yep. That's really cool. Sounds like bees really adapt fast.
They
Is
do
there like is
very
there
fast.
something in their genetics? I don't know if you know anything about it. Is there like something in their
Yeah.
genetics that is able to help with this?
So that's a fun question. So honeybees are absolutely incredible in that the organism in their case, I think, isn't the individual bee, but it is the entire colony, and their biology is kind of very specifically adapted to this lifestyle. So let's talk about their genetics. They have so they have chromosomes like us, and during meiosis, when the cells, reproductive cells, split, for all organisms, you have this genetic crossover. So the chromosomes line up these pairs, and then they swap genetic material between them to just kind of mix it up a bit, and that increases variation, which enables better survivability when the conditions change and stuff. So honeybees have this crossover rate and about 6 times higher than not just any other like animal, but any other eukaryotic organism, and that includes uh, pretty much plants, fungi. I think pretty much anything. Um, so they have this really high genetic crossover rate. They also have this multiple drone mating system. So one queen will choose a lot of different drones, and all of those drones' genetic lines are then in the colony. And we do know there is some segregation in workforce based on patriline, based on who the bee's father was. So some bees are good at some things, other bees are good at other things. And this genetic variation just basically it expands the talents of the entire colony, and allows them to adapt faster. So we covered the crossover, we covered the drones. Now, why do they have all of these things to increase genetic diversity? Well, if you think about the natural state, how they're supposed to be in the wild in Europe, is that I think the natural hive density is about 1 per square kilometer. So in nature, without any sort of influence, there aren't a lot of colonies in an area. And generally, because when a hive swarms, the queen doesn't necessarily go that far. A lot of the hives or a lot of the colonies in one area would technically be very genetically related. So this high variation, this high genetic variation, and these adaptations to increase genetic diversity in the colony is probably a mechanism to reduce genetic inbreeding because of the proximity of related individuals. And that plays very much to their favor when they encounter a new threat, because the diversity then, if you're switching queens around and bringing new material in, um, it gives them a much higher probability that they're going to have an adaptation to this new threat that you can use or that they can use to overcome it.
Yeah. So, say we do find a bee that is able to fight against Varroa on their own without any treatments from us. Is that something we, just with how quickly they adapt, is that something we'll always have to select for, or will that be something they just kind of like hang on to and keep in their genetics?
Well, when you decide to reduce or stop treatments, that is already inputting a selection into the system, right? So the hives that don't have the adaptations to overcome Varroa will tend to do less well. And I think our responsibility as a beekeeper is to find these colonies early and requeen them before they die. But yes, I mean, in terms of, it's just like honey production or how gentle they are. We always need to select for these traits, basically to keep the stock healthy and productive for us. So yeah, the thing about treatment is when you treat a hive, you're removing Varroa, so you're essentially removing the selection pressure of Varroa. So yes, we will have to constantly select for it, or reduce treatments to the point where it's selected for themselves, basically. So where they select themselves.
So, do you think it's even possible for us to eventually find a bee that is able to fight against Varroa?
Yeah, so I mean, people already have in small communities. A lot of them are in Europe. They're managing their bees just fine without mite treatments. I know there's some people in the UK, in Wales, I think specifically. There's a group that's doing resistance breeding, and there's just a lot of individual beekeepers that I know of that are are managing to have these bees treatment free, and a lot of people are trying. So it is possible. Um, I think it's whether how difficult it is probably related to your environment, how many hives there are in proximity, and what other people are doing with their hives. But how it starts usually is you get a few really passionate beekeepers in a small area, and it just kind of radiates out. So these buckfast survivors, it started with this one beekeeper. Now there's a network of beekeepers that have the stock, that breed the stock, that are learning about treatment-free beekeeping, and it's just kind of growing. And it's part of the reason why I was employed here at the Norwegian Beekeepers Association was to, investigate resistance breeding, because that was a very large interest, and it's still growing. So yes, and I think it's possible to get resistant bees in whatever stock you have. I think they have the potential to adapt to Varroa, basically no matter what you start with.
That is really promising because one of the things I've noticed, because I've tried out multiple different genetics of bees in my own apiary, and it seems to be that some bees, I'm guessing it's probably the genetics, you have a little bit of mites in them and they just explode with mites. They have no way of like any sort of resistance. And then you have other colonies where they get the mites and they kind of can like keep them tampered down a little bit for a little bit longer. They might need a little bit of intervention sometimes, but they're showing more promise towards that versus the other ones just also not doing well with like small hive beetles or like other things too. They just seem to be very weak in terms of all the other pests.
Yeah. So there's this variation. I will say with mites, another reason why you want to keep a lid on mites in your apiary is if you decide to stop treating and mites get out of hand, you're actually making it a lot harder for these bees that have a few adaptive traits, but not like the full spectrum or the kind of the full capacity. So you're actually making it a lot harder for them and a lot harder for you to see that they're actually there. Um, but yeah, I mean, even in the control stock that I've been monitoring, there have been 1 to 2 hives that are as good at surviving as the buckfast survivors that I was monitoring. So the traits are there. We just need to make sure we can properly identify them, categorize them, and then select on them, so that we can actually manipulate the population to hopefully adapt a little bit faster than it would be if we just kind of let them die. Because in the Carnica population, we do have a requeening policy. We have this subsidy, so we say if you have a queen that's not really keeping the mites down, then you can get a new queen for a very low price and just change it.
Oh yeah, definitely.
We're working
So
with the queen producer directly.
yeah. So would you suggest any hives in your apiary like that then, to treat them probably before you requeen if they weren't showing any promise towards like
That's what I suggest. Yeah, if you're
yeah.
pretty convinced that this is not a hive that has any sort of adaptation, then I would definitely treat them just to make sure they don't share their mites with everybody else, and then just mark them for a new queen. Because once the genetics change over, they actually can usually deal with the mite problem themselves if you have a resistant queen and you change it.
Okay. Do you think mite bombs are a thing then, since we're on that topic? Where
I
if there's one hive that just spreads to all of them and they go crazy?
So I've got no real kind of data to present to you, but from basic observation as a researcher, yes, I think that is totally a thing. So I think horizontal transfers, so foragers coming back going to the wrong hive, is a thing. But I think also more importantly is if you have a weak hive, they tend to have more Varroa. They also tend to be a target for robbing. And that is a huge transfer of mites. You've got bees from a healthy colony just breaking in and running all over the brood to get to the honey, and they'll pick up mites and bring them back. So my hope is that actually you have colonies that have enough of a resistance that they can actually accommodate this robbing. But it'd be really good just to not have it as a problem, so that you can see your survivors, and so you can give them a little bit of a hand at the beginning if you're just starting from scratch, to kind of build these traits up a bit before you start stressing them out too much with mite bombs.
Yeah, definitely. That's good to know because we definitely had a little bit of that problem this year in our apiary. We have quite a bit condensed into one area, and we had some stronger hives and then some like 5 over 5 nucleus colonies that were strong, but not to the same strength of like a 10/10 kind of colony. And yeah, we saw exactly that. That some hives that had higher mite infestations, they then just started robbing everything. We had a huge problem with robbing this year, so
Yeah.
that's really good to know.
So honeybees are opportunists, and if they see easy food, they're going to go for it.
Oh, definitely. Yeah, they're like teenage boys at a buffet. If they can get it, they're gonna
Exactly.
get it. That's a term that Mike Palmer always used. I thought was really funny.
It's
But
fun.
yeah.
So yeah, I mean, treatments are a tool, and I wouldn't be afraid of using them, and I wouldn't be afraid of employing them to start breeding your resistant stock, because ultimately, it just matters that you're identifying these survivors and requeening the hives that are less good at surviving. I mean, your colonies don't have to die to achieve that. With good monitoring and good beekeeping, you can achieve that without losing your hives.
Yeah, definitely. To go back a little bit, because I had this question but didn't get the chance to ask it. So you didn't mention using like alcohol washes for mite checks. Do you oppose those at all?
So it's a standard research method to do alcohol washes. I mean, I think my aversion to them is personal because I don't like killing bees when I don't have to. Um, so they are more accurate generally than sugar shake or even bottom boards. Why I go for bottom boards is because it aligns with the views of the beekeepers I work with. They don't like killing bees either. They don't like disturbing their bees. Really early in the spring or really late in fall when it's cold and stuff. So I was like, if we need a method that they're going to be comfortable using on their bees. So I was like, all right, bottom boards. You don't even have to open the hive. So it's nice and simple, and we'll see what we can do with that because the breeding program is definitely science based, but it is catered to beekeepers. So it revolves around finding a method that the beekeepers can use to get resistant bees with mitigating loss, and just making it easy for them to do. Because we have some resistant populations that are in kind of the scientific community that they have control of, but the methods for getting these bees are not beekeeper friendly. They're not layman friendly. So, and a lot of the methods that I've been used kind of categorizing the survivability involves me sitting for hours, opening brood cells like one by one in the lab and taking little mites out and counting them. And it's very, very time consuming, labor intensive, and it's just not fun.
Oh yeah, I can imagine so. So
So, yeah.
would you suggest monitoring your mites then like once a month or like every other month, every 3 months? What's kind of like a normal regimen that you like to kind of look at that?
So depending on how fast your mite populations usually grow, so we in Norway with this project I'm running, we do it 3 times a year. We do it spring inspection. We do just when the supers go on, and we do when the supers come off. If you know that there's a high colony density in your area and the mites population grows really fast and can get out of hand really fast, then I recommend once a month. Or if you identify a critical period. For ours, really when the mites start to take off is about mid-July. That's when you can really tell that there's going to be some disasters in your apiary. So then we start, I tell the beekeepers, if you're worried, just start counting, like a couple of times a week in that month, just so you can see if they're really increasing. But yeah, so depending on your mite problem, I would say 3 times a year to once a month, and then more at critical periods if you're worried. And you can mark hives like if they had a very fast growth rate from spring to midsummer. You can keep an eye on them because usually the growth rate for an unselected hive is exponential. So it just kind of it starts here and then it goes wee, it just kind of takes off like a rocket. Um, so getting it when it's just starting to take off and treating the hives, it should kind of put you in a good stead to have that hive survive the winter. But every environment is different. So I'm giving you very blanket terms and advice. And, just cater it to your area. You know your bees, you know your area the best. Um, and it's a best judgment sort of call.
Okay. So did I see correctly that you were also studying bees? I saw Norway, but was there, I think, Sweden and a couple other places were in your paper, or did I
Yeah.
get that wrong?
So
Okay.
continuing the recapping story, when I found this
Yeah.
recapping in the Norwegian bees, I was like, "Whoa, cool!" So then I went to my colleagues in Sweden and France, and I'd read all their papers, and I was like, "Hey, you guys have resistant bees. Do you want to look for this? Can you just look for this?" So we did a collaborative paper, which was published in 2018 in Scientific Reports, because they all looked for this recapping behavior in their own hives, and they found it. So we had then two populations in France. We had one in Sweden and one in Norway that had nothing to do with each other genetically. They were completely isolated from each other and yet had the same trait at really high levels, much higher than the controls, and it was much higher in cells that we'd found mites in, this recapping behavior. So it was kind of a convergent trait. We had four individual populations that were that didn't kind of have any genetic mixing whatsoever, had this trait, all of them together, and much higher than the controls in the same geographical regions. So that was the first evidence I had that recapping was going to be highly associated with survivability and mite surviving bees. It's a pretty cool paper. I can also send you a link to that. It's open access, so you can read it.
Yeah, definitely. I'll put them all in the description for sure. A lot of my followers like reading all the articles and staying up to date on all of that. So yeah, that'd be perfect. So were those all like buckfast bees, or were they other different breeds of bees? Because you also mentioned carnolians.
Yeah, what I call Carnica bees. Um, so we're working with a Carnica stock here in Norway. I think most of the bees that had these adaptive traits were just kind of mixes of everything, just the general bee that was in the area. The Avignon population in France was actually from an abandoned apiary that one of the researchers stumbled across, found these bees there, and were like, those should be dead, but they're not. And then kind of bred from the stock and saw what was going on. And in Sweden, there was an experiment called the Bond experiment, live and let die, where they found an island called Gotland. This researcher, Ingmar Fries, took a bunch of bees, put them on the island, and left them there. And the same thing happened. A lot of them died, and they kind of developed resistance slowly. Now, I mean, the live and let die method seems to be fairly popular. I don't like it so much. One, because it's not very fair to the bees, but also two is because if they're allowed to requeen and manage themselves, they tend to get smaller, they tend to get more aggressive, and they're not really good for beekeeping. So as far as I know, the Avignon and the Swedish populations aren't being cultivated for commercial use because they're not competitive. Whereas the buckfast population, because it was created by a beekeeper, it produced competitive levels of honey. They were very gentle, very calm on the frames, and very easy to work with. I love working
Hmm.
with the buckfast. Honestly, they're so nice.
Okay. So it is possible to select for more than one thing then in your breeding program. You don't have to just solely focus on only Varroa resistance. You can focus on other things too at the same time.
Oh yeah, definitely. And I encourage people to do that because you don't want to lose traits that would make them easy to work with as domestic bees. You need the gentle stock. You need them to be calm. You need them to be highly productive if honey is your thing. Um, Kind of those traits are things that cannot be lost because then what happens if you'll have a stock that is not very usable for be resistant, but it won't be very workable.
Yep. Yep, exactly. All right. Well, this is all so cool. Thank you so much for sharing all of this with me.
I'm so happy to
So
share it.
yeah, definitely. Definitely want to start getting some more information into the beekeeping community about the progress we're making with Varroa mites and bees. Because in America, it seems to just be, like I mentioned before we started, just kind of like a cookie cutter of like, this is what you need to do. Don't really think outside the box. But it's cool to see new people starting to push outside that.
Oh yeah, I do recommend thinking outside the box. I mean, definitely be careful because there is risk associated with going treatment free. Always, it's never not there. But do think outside the box. Do read things outside of the United States. There's a lot going on in Europe, in a lot of other countries that have much higher colony densities than Norway. They're working with it. They've got a lot of ideas. Reach out to people. A lot of the time, scientists their emails are on the papers. that are published open access, so you can approach them. Try and communicate, talk with other beekeeping communities. I know there's a Varroa resistance website that just went up in the UK, run by Professor Stephen Martin, who is the guy on the Hawaii paper with deformed wing virus, the variants that we talked about. So there's a lot of resources. There's a lot of beekeepers into it. Just start talking about it.
Yeah, definitely. Well, before we hop off, where else can we find you and follow your work?
Right. So I'm on ResearchGate, researchgate.com, I believe. It's kind of like Facebook for researchers. You can put a link there if you want, with my permission. Also, if you look up my profile on Google Scholar, you'll find a list of all of the publications that I've been involved in. And if I'm corresponding author, my email will be on those papers if you want to approach me. I have kind of a high volume of emails, so I might not be able to answer everybody, but I'd be very happy to give it a shot to try. So if you have a question or you really want to talk to me, then just send me an email.
Okay, awesome. Well, thank you so much for chatting with me.
Thank you very much for inviting me to talk. It's always fun to talk about bees.